mTORC1 is also very frequently activated in the most common types of human malignancy, further making this question a very important one in the field of cancer metabolism. You can see that the pathways that lie upstream of mTORC1 are comprised of common oncogenes such as PI3 kinase and RAS, as well as tumor suppressors including NF1, and the TSC1-TSC2 complex itself is a tumor suppressor. So we wanted to ask whether in cancer settings where mTORC1 is activated, whether mTORC1 is required for driving de novo lipid synthesis. The first place we decided to look was in the kidney tumors of TSC2 heterozygous mice. These mice, when they lose the wild-type copy of TSC2, develops renal cyst adenomas. You can see these pictured here, and this is staining for phospho-S6 as an indicator of mTORC1 activation. You can see that the tumors stain very strongly for phospho-S6 compared to the surrounding normal tissue. I looked at expression of SRBP targets in these tumors and found that they very nicely correlated with mTORC1 signaling, where expression of ACC, fatty acid synthase, and G6PD are all elevated in the tumor compared to the surrounding normal tissue. To ask whether or not these increased expression of these targets is due to activation of mTORC1 signaling, we treated mice with rapamycin or vehicle, and what you can see is that in vehicle-treated animals, elevated expression of fatty acid synthase and ACC are found in the tumor compared to normal tissue. However, when you treat with rapamycin, just 2 doses for 36 hours, you can see that this increased expression is now gone, demonstrating that in vivo, in these mTORC1-driven tumors, mTORC1 is controlling expression of lipogenic genes. Further studies will be done in collaboration with Stefan in the lab to determine whether these effects are through activation of processed SRBP and ultimately effects on the transcript and protein levels, doing Western blot and RNA analysis from tumor tissue from these mice. We were fortunate enough to collaborate with Chin-Li Wu at MGH to also look at patient samples from TSC patients, which is the genetic tumor syndrome that arises when patients are heterozygous for TSC2. They will lose the wild-type copy of TSC2 like the mice and get a variety of lesions. One of the most frequently found is angiomyolipomas, which are very vascular tumors that also have smooth muscle and adipose components. Chin-Li Wu stained for fatty acid synthase, and what you can see is in these 2 patient samples, there's very strong staining for fatty acid synthase in both the smooth muscle and in the fat components. And this is a normal fat staining. For comparison, a normal fat actually does express fatty acid synthase, further demonstrating that there are very high levels of expression in these tumor samples. And future studies will be done to perform these types of analyses with additional antibodies for SRBP targets on a microarray of samples. In order to look in contexts that are more relevant to human malignancies, we used lung tumor samples kindly provided by Jeff Engelman and Kwok Wong that are driven by oncogenic mutations in PIK3CA and KRAS. We found that staining for fatty acid synthase and G6PD, 2 SRBP targets, were very strongly elevated in the tumor tissue in these lung samples. We want to do further analysis in order to determine whether rapamycin treatment of these lung tumors reduces expression of SRBP, and that is a future direction for this work. However, we had a cell-based system to address this in. Marisa Binden in the lab created NCF10A lines that she transformed with either a vector, activating mutation in PIK3CA, or activating mutation in KRAS, to ask whether these oncogenes can drive lipogenesis in a TOR-dependent manner. I measured de novo lipogenesis in these cells. What you can see is that activating mutations in both PIK3CA and KRAS strongly induce levels of de novo lipogenesis. However, this is partially suppressed when you treat with rapamycin, and I've done further studies to show that treatment with catalytic domain inhibitors of mTOR1, of mTOR, such as Torin and PP242, suppress this even further and really bring it down to wild-type levels. To confirm that de novo lipogenesis downstream of these oncogenes is due to SRBP, we've shown that knockdown of SRBP 1 and 2 also suppresses the oncogene-induced levels of de novo lipid synthesis in this setting. Stefan is following up on these studies to further understand the contributions of mTORC1 downstream of these oncogenes in the activation of lipogenic gene expression and de novo lipogenesis. Finally, I want to share with you a collaboration that was done with Karen Chakowski's lab. Here they have NF1, which is a RAS-gap deficient malignant peripheral nerve sheath tumors. When you lose NF1 tumor suppressor as well as P53, malignant peripheral nerve sheath tumors form, as well as the same type of model was done with lung cancer. And what you can see is that they can treat these tumors, which are traditionally very hard to treat, with a combination therapy, including both rapamycin and an HSP90 inhibitor, IPI-504. And this combination therapy produces a very pronounced response and completely eliminates these tumors. We hypothesized that mTORC1's control of metabolism might play a role. The HSP90 inhibitor is known to promote ROS, and mTORC1 is known to promote G6PD activation, which can produce NADPH to help ameliorate this ROS. So we hypothesized that inhibition of both mTORC1 and exacerbation of ROS with the HSP90 inhibitor might be the mechanism whereby these tumor cells are driven into cell death. And we were able to show that treatment with rapamycin in these tumors indeed blocks G6PD expression as well as other lipogenic targets and are mechanistically involved in the cell-killing phenotype of this combination therapy. So finally, this work begins to scratch the surface on understanding how oncogenes and tumor suppressors that lie upstream of mTORC1 might regulate specific aspects of cellular metabolism. We're specifically excited about SRBP and further understanding how targeting this cellular metabolic pathway may have therapeutic potential. So finally, just to summarize, mTORC1 can activate SRBP to promote de novo lipogenesis as well as the pentose-phosphate pathway in cells, and this seems to be important, although the regulation is slightly different in metabolic tissues and in tumors. So with that, I'd like to thank you for your attention. I'd also like to quickly make some acknowledgments, and I know I want to leave some time for questions. I'd like to thank the Manning Lab. I will save my thoughts and comments on Brendan as an advisor towards the end, but I would like to thank Sue for her tremendous help in all of the mouse studies that I did, and working very closely with me on the cell metabolism paper. And I'd also like to recognize Katrin and Hui, who both began both of the projects that I worked on and were very helpful in setting me up to accomplish two great projects in my thesis PhD. I'd like to thank our collaborators for reagents and technical help as well as advice. And I'd like to thank my dissertation advisory committee, Louis Cantley, Jihao Li, and Gokhan Hood-Mislagil for their guidance and support and advice, as well as the members of my dissertation defense committee, Gokhan, Tiffany Wong, Marianne Wessling-Resnick, and David Sabatini, who came from MIT. I would like to quickly remember my friend Peter Curtin. The three of us, this is my husband Derek, we're supposed to leave Boston with our PhDs, and he is unfortunately no longer with us, so I would like to remember him today. I'd like to thank my parents and brother, Justin. They're also here today, as they always are for me, have been very supportive, particularly my mom, who inspired my passion for science and really was an excellent role model for being passionate about what you do. I'd also like to, as Brendan mentioned, I did get married and have acquired a new family in my PhD, and I'd like to be thankful for them. I'd also like to specifically thank my mother-in-law, Laura, and my grandmother, Maxine, who came to be here today. And Derek, this picture of Derek and I and Brendan also showed, but it's actually very funny, especially because Derek is from California, and I also have to thank this coat for having him come to Boston and be able to bear the winters with me, and be very, very supportive in everything that I've done. I definitely couldn't have done it without him, as well as his handiwork in the lab. He worked for me as a technician, so I really couldn't have done it. I'd lastly like to thank Brendan. Brendan has been a terrific advisor, so supportive, and really it wasn't such a difficult decision for me deciding between the two labs. I actually was going to bring up the same story. Brendan gave me so many projects and exciting opportunities, and it's very typical of Brendan that he has so many